So we heard this morning AI is actually sort of old news, but we have a really young revolution going on, and which is driven quite often by young people. And so we decided, therefore, we're going to invite also a young Texas AI researcher. I'm very pleased to welcome Dr. Zhou from the Department of Bioinformatics at UT Southwestern in Dallas.
Hello, everyone. I'm very honored to have this opportunity to share with you about my research. So I'm still learning to use this. Most of my career has been dedicated to the problem of understanding the genome through the lens of AI. So my journey actually started from developing one of the earliest deep learning models for the genomic sequence. And one of my major reasons for working on the problem of genomic sequence over the past decade has been that I think the genomic sequence could be the most underexploited resource in genomics. Right? So this might sound surprising because genomics is a study of the genome, right? But most of the time, we're actually using genomic sequence as a tool as a reference to make a measurement or detect mutations. But the information that's actually encoded in the genomic sequence, or the biology encoded in the genomic sequence, is actually rarely extracted as a source for driving scientific discovery. So that's a potential untapped opportunity, right? And this, of course, is because it's still hard for us to understand the genomic sequence and its impact, right? So understanding the genomic sequence is a problem that is quite challenging with traditional biology approach. I would argue that it's a problem that's almost perfectly set up for machine learning and AI. Because first, we have the entire human genome. We have 3 billion base pairs. And at the same time, we have tens of thousands of data sets that measures different types of genomic sequence properties or activities. Right? So, which makes the genomic sequence probably one of the most well-annotated data sets that has ever existed. However, just having those external data won't directly tell us how do those activities depend upon the genomic sequence. So for example, if it takes the famous example of AlphaFold. So having the protein structure and protein sequence alone don't directly tell us how does the structure depend on the sequence, right? But this dependency can be inferred through computational modeling approach through machine learning and AI, right? And the same applies for all the rest of those genomic sequence activities that we study. So I think there is a huge space here for potentially developing AI and machine learning techniques to capture and exploit the link between sequence and its activities. So we take a global view. I think there are 3 main directions where AI can contribute to the genome. And I call it prediction, understanding, and design. And I link it to the famous AI applications that's relevant to each direction. And for understanding, because the current AI algorithms are still not great at extracting simple principles from complex phenomena, we still need to rely on the human intelligence. And it's likely an important direction for developing future AI for science. Right? And I will go over each one of those directions to give you a concrete example to illustrate the problem that we can solve in each direction. So I'll start with prediction, which is one of the most developed directions. And just like with AlphaFold, we can predict from the protein sequence to protein structure. We can develop those predictive AI approaches that can predict from genomic sequence to various types of genomic sequence activities. And currently, one of the major challenges in the field is that how do we effectively make use of distal sequence information? Or in other words, how do we scale the model effectively to use longer and longer genomic sequence to make better prediction? And when we are looking at genomic sequence that's beyond a few kilobase pair, one of the properties that's important and we cannot ignore is that the sequence is not just a linear sequence that's existing in the nucleus. Right? It actually has a highly spatially organized structure, and it has different characteristics at different spatial scales. So we look at very local scale versus, we look at very local region versus we look at it much larger, region, we can see very different distinct characteristics, right? And this multi-scale 3D genome organization can be well measured experimentally through techniques such as Hi-C and represented as those interaction matrices on the top, right? So in those interaction matrices, each pixel indicates interaction strengths between a pair of genomic locations. Like in the leftmost plot, the white dot here indicates a chromatin loop between a pair of genomic locations. And we can have this measurement at very different scales, from very local to entire chromosomes, right? So to be able to model the genomic sequence at very large scale, we first need to be able to capture how the sequence encodes those 3D genome organization. So that's what we want to do. We want to be able to predict from the genomic sequence to the entire multi-scale organization that we can measure. So this presents us with a actually unprecedented challenge, which is the size of the problem. So we are looking at the entire human chromosome. It can be up to 200 million base pairs long, right? And that is a single input. So it is much larger than any other deep learning applications that we have known. And on top of that, if we're considering the interaction matrices, that's of size square of the length, right? Which can make the problem even more intractable. So how do we solve this? So fortunately, the genome interaction data can be naturally represented in a hierarchical way. So I like to make the analogy of a Google map. Right? So if we look at a very large region, for example, a whole chromosome, we're mostly looking at long-range interactions, and those interactions are only measured at lower resolution. So it's sufficient to represent those large regions in lower resolution. And then as we iteratively zoom more and more into smaller regions toward our position of interest, we can represent those interactions in higher and higher resolution, right? So that's an approach that we're building into the model, and combined with some other scaling techniques, we're able to scale the model to make predictions for genomic sequence that's longer than an entire chromosome. And it's shown in an example here. So we start at the bottom right, starting from prediction for more than an entire chromosome. And then we iteratively zoom into smaller and smaller subregions of that chromosome, and all the way until we reach the smallest region where we can make predictions at the highest resolution the data allows. So importantly, here, even though that we are making prediction for a chromosome that the model has never seen before during training, we can show that the prediction actually aligns very well with the experimental measurement. So now what can we do with this capability? So one of the major applications is that we can now use it to predict the effect of any mutations. So both the mutations that we know and mutations that we have never seen before. Right? So for example, here, we're looking at a structural variant that is a deletion on a chromosome that will cause this hand development disorder. Right? So now we can take this model and make predictions for the 3D genome structure before and after the deletion. Right? And our prediction shows that this deletion actually causes a new interaction between the PAX3 gene and the enhancer. Right? And this turned out to be the cause of this disease. So in all cases where we tested our models on structural variants that we have known experimental data for 3D genome organization, the prediction actually aligns very well with the experiment. So I think there is a real potential here that we can apply these predictive models to the vast majority of the structural variants that we have no experimental data for, and help understanding their association with disease. So the predictive modeling algorithms give us a natural question, which is, how does it do that? Right? So in many of the cases, the model's predictive capabilities seems to be beyond our current understanding about the sequence-based mechanisms. Right? So what can we learn from those models? So predictive models are very useful tools for answering this question, but at the same time, there are also black box models that don't directly allow us to look into it to understand its inner workings. So we still need to develop a new approach for us to gain a better understanding about the sequence-based mechanisms. So here I'll show you an example that we developed an approach to look into a basic question, which is what defines a promoter at the sequence level. So we know that promoter is one of the most essential element of a gene, right? The promoter is responsible for driving the initiation of transcription of the DNA into the RNA. So all genes have to have a promoter, right? Because without promoter, it won't be transcribed and it's not a gene. So despite that promoter is having this fundamental role in the genome, so we are asking the question of what should the promoter look like in the human genome? Actually, our answer is still quite incomplete. So over the past few decades, the field has identified a couple of core promoter motifs across many different organisms that are shown here. So motifs are like words of the DNA. And for average human promoter, you can still have none of those known motifs. So for example, the most canonical promoter motif, TATA, is TATAA. It exists only in about 10% of human promoters. So for most human promoters, actually, if we just look at the sequence itself, it's still hard to understand why is it a promoter and why the transcription starts at the position that it starts at. Right? So we want to tackle this question by first formulating it as a machine learning problem. So the problem is that we start from the genomic sequence, and we want to predict the transcription initiation signal at the base pair resolution, which are experimentally measured. And we first developed a deep learning algorithm that can do this prediction well. And importantly, we're able to analyze that deep learning model and extract how does it do the prediction and use that understanding to build a simple model that have very few parameters, and we can explain every step of its computation. And it still does have achieved similar performance compared to the deep learning model. So how does it do that? So the core of this simple model are the motifs and their effects on transcription initiation that it has learned. So the motifs are completely learned from just the sequence and transcription initiation data, where you see that we recapitulated some of the known canonical promoter motifs like TATA and several other common promoter motifs. And importantly, most of those transcription factor, most of the motifs that we identified, even though that they match known transcription factor motifs, so their effects on transcription initiation was not clearly understood before. And those are shown on the left plot. So left plot shows the effect curves of each motif on transcription initiation. So how to read it is that we put the motif at the center of the curve that's indicated by the vertical line. So the TATA motif will activate transcription at almost exactly 30 base pair downstream of the motif. And Y1, in contrast, the motif will activate transcription immediately upstream of the motif, and also about 200 base pair upstream of the motif. And next, we see a bunch of those motifs that have bidirectional effects on both strands of the DNA. So you activate transcription bidirectionally on both directions at the same time, which were not previously known. And they're likely the basis of why most human promoters have bidirectional transcription initiation. Right? And if we look at more closely in each of those curves, we actually see each one of them has some distinct characteristics. So for example, the FY motif's effect has this 10.5 base pair periodicity. So 10.5 base pair happens to be the helical periodicity of DNA. So that likely indicates that the NFI transfactor might have a more rigid interaction with the PO2 pre-initiation complex, which dictates that the transfactor might have to bind at a certain surface of the DNA. So I think each one of those curves here likely reflects the underlying mechanism of how they activate transcription initiation. And of course, that we can also experimentally validate the effects that's estimated by our model. So for example, we can ask the model what would happen if we remove one of these transcription factors. So because the model is fully explainable, if we want to simulate the effect of removing the transcription factor NFY, we can just remove the effects of NFY from the model. And the model will make prediction about what's the effect of depleting NFY on the transcription initiation signal. And those are shown, the predictions are shown on the left plot, and which agrees almost exactly with the experimental measurement of depleting NFY through knockdown. Right? And again, in this case, the model has never been trained on this data set, but it can accurately make predictions about what will be the effect of removing a transcription factor. So fast forward, now we are having a model that can actually now use with a simple model to explain most of the human promoters, right? And moreover, for every one of those promoters, we can explain what are the base pairs and motifs that contribute to the activity of those promoters. So this allows us to further study what are the connections between the promoter base pair and motif level contribution and the function of the gene, right, which I won't go further into. I want to emphasize that promoter is perhaps still the most simple and well understood type of regulatory element in the in the genome. And there are still many different types of regulatory activities that we know even less about what's the sequence level mechanism. And this approach, we can potentially generalize it to apply to study the sequence basis of all those different genome regulatory activities. So finally, I would like to talk about the direction of design. So design is the flip side of prediction, right? So instead of making a prediction from the sequence to its activities, now we want to ask, so we want a particular type of activity. For example, if we want to drive gene expression in a particular type of immune cells, or a particular type of tumor cells, how do we design the sequence to give us the desired activity, right? So this potentially can have applications in not just basic science, but potentially also in bioengineering or even medicine, such as gene therapy in the long term, right? And to enable that, we also take some inspirations from the generative AI approaches for images, right, which can do amazing things, like given some text instructions, you can generate images that has never existed before, like an astronaut riding a horse on Mars, right? So we want to use this capability for designing sequences that might have activities that we have never observed in the genome, right? So how do we do that? So those generative AI techniques are built upon a method called diffusion models, which are actually based upon the foundational work from Dr. Ganguly's lab, who's our next speaker. So we want to generalize the diffusion models toward designing genomic sequences. So without going into the technical detail, so the intuition of diffusion model is that by learning to denoise a noisy image, for example, if you ask a neural network to denoise a noisy dog image, the model needs to learn about the concept of a dog, or what a dog should look like, right? So similarly, if we ask the model to denoise a promoter sequence, so the model could learn what a promoter sequence should look like, right? But the original diffusion model were more suitable for generating continuous data, like the images, but not suitable for categorical data like sequence. So we developed a new diffusion process that's more suitable for those 0-1 encodings that can be easily used to encode genomic sequences, right? So with this model, we can apply it to generation of categorical data, but before we apply it to generation of sequence, we actually first apply it to generate another type of categorical data, which is Sudoku. So why Sudoku? Because it's also a kind of highly structured data that need to satisfy hard constraints. So we want to test our model's ability to generate this kind of highly structured data before we apply it to the problem of generating sequence, right? And plus, that to our knowledge, this problem has not been solved before with the generative model. So what we do is that we feed the model with those fully filled pseudocode examples and without telling it the rules. Right? And it turns out the model learns to generate pretty much 100% correct pseudocodes. And moreover, a model learns to solve pseudocodes without further training. So by conditioning on those digits that are given and generate the rest, the model can solve most of the pseudocodes just in one try. And even for the hardest examples, if we give it multiple trials, it can also solve them. So now we know that the model has this capability of generating those highly structured data satisfy strong constraints, and we apply it to the problem of generating sequence. So here, we're looking at the inverse problem of the problem that we just talked about, which is if we are given a transcription initiation signal of a promoter, what should the underlying sequence look like? Right? So we can see that the model design sequences shared many characteristics as the ground truth underlying true promoter sequence. And moreover, we can now also experimentally show that the design sequences can actually give us the activities that we ask it to design for. Right? So I think this capability can potentially be generalized for us to design sequences for all kinds of different genome regulatory activities. Right? So finally, so I've shown you all three of those directions. And I think all of them are still in an early stage, and there are still a huge potential for future improvement in all of them. So especially for the direction of understanding and design, I think we're really just getting started. And there are also still a lot of untapped synergies between those directions. Like if we have a better understanding about the process, can we build that understanding into our model to build better predictive and design models? Right? So if you have built better predictive and design models, we can analyze that to gain a better understanding about the underlying sequence-based mechanism. Right? And the prediction and design models can be synergistic to each other too. So I think there is really a huge potential of using AI to improve in all three of those directions and potentially help us tackle the question that we posed at the beginning, which is really to unlock the information in the genomic sequence to drive biological discoveries. So finally, I would like to thank my team who did this work and my funding sources, and thank you for listening.
Thank you, John, for a fascinating ride. Please, questions.
Yeah, thanks. Richard Gibbs, Baylor College of Medicine. My question is about more data or more models. We have a lot of genomic data, and we're making more, mainly in the context of disease gene discovery. But for the purposes of understanding things like you're talking about today, the repertoire of promoters, do we simply need more data, and what kind?
So in the question of data, I think on the one hand, we are kind of lucky that the human genome already provided us with quite a lot of data, the 3 billion base pairs. But at the same time, we are also limited by our genome size and its variations, right? So the 3 billion base pair is still not infinite amount of data, which might not allow us to answer all of the questions that we want to answer, right? So potentially, to get to when we reach, I don't think we have reached the full potential of the current data yet, but if we reach that point, I think maybe the only way to improve through the machine learning approach is to be able to generate new data, like being able to synthesize sequences, put them into cells, and test what are the effects, and use that to increase the amount of data.
Okay, thanks.
Please.
So this was a very nice talk. I have a couple of curiosity basically, particularly the problem of chromosome structure that's dear to my heart. You're looking at high C data, most of it, and but your predictions are only in 2D. Don't we have to go through the three-dimensional structure to actually go forward? Based all your comparison appears to be only in 2D, and then how we can learn something and figure out from those patterns. Do we have some underlying rules that we have learned, or just to say do we have prediction power?
Yeah, that's a very good question. So first question is that our prediction is in 2D, right? So also, even though I think it reflects the kind of ensemble of 3D structures, right?
It's a million cells in high CIEL, right?
It's not directly giving us what's the 3D structure in individual cells. I think it's a fascinating question if we can actually move beyond from predicting an ensemble average structure, but predicting individual 3D genome structure that might occur in individual cells. So that might be a very, I think, important future direction, right? So the second question is...
No, but the first question, there are a lot of people like us and other people doing these 3D predictions. So it'll be interesting to compare where we'll go compared to what already exists.
Yeah, so there are a lot of methods that can go from the 2D maps towards the reconstructing the 3D structure, right? So, yeah.
Ensemble of structures.
Yes, an ensemble of structures. Yeah, exactly. So, yeah, I think we just want to maybe just move from connect that to the sequence-based prediction as well, right? So from the sequence, we can predict an ensemble of the 3D structure. Yeah, that would be something that would be really fascinating.
Okay,
yeah,
very nice. Thank you.
Yeah, sorry. And for the second question of understanding the prediction models, right? So actually, we also did some analysis to understand how our model are making the predictions. So one of the things that the model enable predicting is that we can predict those chromatin compartments, like the genomic DNA or the chromatin can be separated into A and B compartment, right? So we ask what are the sequences that drive the compartment. And by doing some computational experiment with our model, it turns out that, at least according to the model, the main driver for A compartment is the promoters, the transcription start sites. And even just very short transcription start site sequence, like 400 base pairs, is sufficient to establish A compartment. But to establish B compartment, we need an extended chunk of DNA sequence that does not have promoter activity. Like at least 10 kb of those sequences will be needed to drive the formation of B compartment, which also matches with some of the recent experimental measurement. So I think that is still more of a hypothesis or model that's generated by those predictive AI models. But it will be interesting to see that whether it will be validated through experiment in the future.
Let's take one more question over there.
So you described beautifully how to use machine learning to try to understand promoters. But we came up with the concept of promoters in an attempt to understand transcription. What would happen if we just imagine 30 years ago, just through machine learning at transcriptions? We will never have even the concept of promoters. So I'm worried that it's very easy now to throw machine learning. I think it may come at the expense of understanding. I don't think we would have the concept of promotion. In fact, we may not even have a concept of a 2D sequence, which is just throw machine learning at the whole cellular data. Let's see what happens.
So I would say I think there are it's certainly true that for machine learning approach, sometimes we oversimplify things into just prediction, into the input and the prediction, right? But what we want to be able to achieve here is be able to actually extract some information that are not previously known. So for example, from the model, because we have built this experimental model, we are now able to actually know a lot more about the motif effect on transcription initiation that we previously didn't know about, right? For example, how does the Y1 motif affect the transcription initiation? And we have not have an answer that can also be experimentally validated. So I do think there is a value in developing those machine learning approaches, but more importantly, to make it something that can allow us to understand it and use it to gain new biology. So yeah, I don't know if that answers the question.
Thank you.
Well, I think that exchange is exactly what we wanted to accomplish with this session. That, I think, is the core question, right? So let's thank Jian again for a great talk.